So I've got a ton of questions, but does anybody? Well,
Louisa, you're working on your doctorate, right? Or
she should be, but no, she's just.
like with our gene, we're going to be Dr. Louisa.
Yeah. I'll get there.
Okay.
I will get there. I am an international student in this country, but I will get there one day. Yes.
Don't you be deported. So quick question. Brains are a variety of cells.
Yes.
And so you just talked about the neurons. What happens as far as astrocytes and other microglia?
This is something that we can definitely get a glimpse on through the bulk RNA sequencing as well, because in there we're getting RNA, we're getting transcripts from all of the cell types.
Sure.
And in here, actually, this little thing is a very, very, very simple diagram of what that neurorosette looks like. And we already see like different cell types that you can recognize. So in the red and the yellow here, we have stem cells. And then these blue things, these are actually radioglia, which arise spontaneously in the organoids as well. So these are glia. And then at the end, we also have astrocytes. So you have all these different cellular types that show up in the organoids and that we can work with as well.
Okay. Oh, okay, I'm standing in the wrong spot. Okay. And so they just develop normally, those other cells. How long can an organoid live?
We celebrated the second birthday of an organoid batch last year. So they can grow. As long as, they can grow. They don't grow in diameter as much because they're not vascularized. They don't have any blood going through them. And so at some point they start generating this necrotic center where the nutrients can't get there anymore. And so they're about 1 to 2 millimeters in size, but that, they're big and we can see them to the naked eye and they're like my pets.
And it's kind of cool, but kind of creepy. Well, do they deploy? Is there natural neuronal activity?
A lot of spontaneous activity, and as they grow, we see that the activity also matures. And so first you see a lot of spikes, so a lot of firing rates, very high firing rates. But as they evolve, you start seeing some synchronicity and some rhythm to the bursts.
Do they look like they're thinking? No.
They might.
Okay. Say that. Okay.
Ladies first.
Okay, ladies first. No, ladies.
I just want to. I think the splice variant. Are they the same splice variant?
Yes.
Yeah. I think that this is +5 and +4 and 5, and it's probably in frame. I think it is very good you are going to check with RT-PCR. And if you are going to do it, I would recommend that you use both with NMDA inhibitors, nonsense-mediated decay inhibitors, and with and without.
Okay.
And if you want protocol, I'll be very happy to share my protocol with you. So I think it's very important. Yes.
I'm going to write it down.
It's an in-frame deletion of exon 2, and it results in a reduction.
It should be, because it's within the...
Yeah. Exon 2 in frame reduces by 3 or 4 kilodaltons. So if you do a long Western, you might see a stable, shortened form. And RT-PCR. Yeah. Okay, enough of us talking.
Quickly ask, what was the time point for your RNA sequencing?
Time point.
Yes. How old are they?
They were 3 months old.
3 months old? Okay. Thank you.
Rami has a question. Just go down the road there.
Well, thank you very much for a very nice presentation, and same for Sharon. Everybody. Wanted to ask actually a similar question when you looked at the staining, the immunohistochemistry in the organoids. What age were there, and where did you compare different ages?
Yeah, so they were 3 months old. We also cultured them up until 5 months old, but I haven't done that analysis yet. That's okay. Yeah.
And can you explain us the rationale why you did RNA sequencing? I mean, for a postsynaptic protein. Given. Yeah, I know,
but
we still want to challenge here. So.
Yeah.
And
So
given that especially that when you take the organoids, you look at different cell types. So you have those RGs, you have those neurons, you have those astrocytes. So you will never know exactly where the signal coming from.
Yeah, that is
Right.
correct. We don't know. We don't know if it's coming from the astrocytes or if it's coming from the neurons. We do know that the neurons are the most abundant cell type in the organoids, so that that is already a good, it gives us a first impression. But the RNA sequencing, we performed it because it was the first plan that we had ahead. But to be honest, what I really want to do with these samples is a proteomics analysis. I think that would be much more interesting. We would be able to catch those isoforms much better. Because even for the analysis of the RNA sequencing, sometimes depending on the algorithm that you use or the bioinformatics that is helping you out with it, they might not be able to catch different isoforms as in the RNA form. So the proteomics would be a better analysis.
In the bulk RNA sequencing, did you see DLG4 transcript differences?
Yeah, so like I'm mentioning to you, I got this week. So I am going to look, that is the first thing I want to look for exactly, but these graphs were the first thing that I received, and I asked just for a preliminary analysis, and that's what I got. So, yeah.
Okay, go ahead.
Can I ask one more question really quickly? Are you planning to stain for PSD95 along with some other cell type markers, like for astrocytes or for, these are 3 months old, they should be expressing some astrocyte markers, or no, you don't see anything?
They start to. At 3 months old,
okay.
they start to. Ideally, at the 5 months old, I would see a higher expression.
Do you see PSD95 earlier, like around 30 days in vitro? No?
No.
Okay.
I don't.
In cortical organoids? No.
No.
And these cortical organoid protocol is the PASCA protocol, the Beery et al.?
No, it's the Mochi
It's the
protocol.
Mochi protocol, of course.
Yes,
Yes.
which is a less guided differentiation protocol,
see.
so they might take a little bit longer to mature. That could be true. But no, we don't see PSD95 up until we have fully differentiated neurons.
That's very interesting because I have some data which shows 30 days in vitro in cortical.
Yeah?
Wow.
Cool.
Okay, cool. If you got mouse brains given to you from Sharon, could you grow organoids out of those as well?
Mouse brains?
Yeah. And a whole different area, or is that?
No, that's pretty similar.
Okay. Okay. Okay, cool. Just as far as the resolution of the antibody, and here, I'll give you this, and I'll talk from the microphone. So part of the problem with the resolution of these large, bulky antibodies is that you have to put a secondary antibody on. Gerardo just got the nanobody PSD95. Have you guys looked at that at all? Or has anybody in the room looked at that, which is almost like a single chain tagged antibody? Gerardo, is that the one that we're using? Yeah.
Single chain. Yes, as Alex just said, it is a single chain, and it's also coming from a different host. It uses a llama that actually it's a lower spectrum for attaching to the PSD95 that makes it more specific. So we've been looking at the results right now, and we're getting on our immunofluorescence a sharper image for PSD95 with less background, and even the way that it attaches into the neurons on the dendritic side and the nuclei, it looks significantly better than the other ones that we've been trying before.
Yeah, yeah. Great. Yeah, okay. Oh, yes. Here, I'll take it and walk it over. I'll pretend I'm Zainab. Oh, what's that? Oh, okay. You're allowed to talk. We'll come up with a question. Okay. Save me here.
I wanted to ask Sharon regarding the mice. Have you looked at different ages when you did your behavioral tests, like comparing with aging? I mean, we know the mice are not human, but sometimes the onset of the phenotypes comes at a later stage. So whether you compare that.
So what we see in our mice is, for a lot of these tests, we can't actually do them when the mice are really young just because they're really small. So they're too small to actually do many of the tests. But when we look at sort of early adulthood versus late adulthood, we see a lot of the symptoms actually seem to improve in late adulthood, so the symptoms are less severe. The mice are better at performing the tasks. But most of the tests that you saw up there are sort of in the early adulthood of the mice, so when the mice are somewhere between 2 and 4 months old.
Okay, and a follow-up question.
Sorry,
Sorry, just,
microphone.
so probably there could be some redundancy in the postsynaptic density proteins that could overcome those phenotypes. And the question is whether you did or you plan to test for other proteins like DLG3, DLG2, to see whether this really compensate in the mouse at least. And I don't know if you did at all staining for PSD95 in the brains of these mice to show that it is haploinsufficient as you showed in the RNA level.
Right, so...
Or the protein, even the Western, the very nice Western.
Yes, we had a project going on doing the staining. It's all technical stuff is the reason why that's all been delayed. You know, pathology core doesn't know how to cut brain slices, like all kinds of like little things like that that just have been delaying that. But that is something that's in the works. And what was your other question before that?
So
Sorry.
redundancy,
Oh,
like
yes.
mice doesn't show the phenotype as the human, right?
Right, and what we've got in the works right now is looking at doing some just like old school microarray slides, looking at a bunch of different synaptic proteins and looking at the difference in their expression in the shine mice versus the control. So that's in the works. It's a great question.
Right. And that speaks to a question of genetic modifiers. If you look at different mouse strains as far as the severity of the DLG4 knockout or haploinsufficiency, it really changes, making us wonder about there being other genes which can modify the severity. And the obvious question becomes, for humans, is that the case as well? That not only is it the DLG4 mutation, but there might be another 1 or 2 or more genes in the background making it milder or more severe. And that can be useful biological information, also therapeutically, when one looks at diseases such as Duchenne muscular dystrophy with utrophin and other diseases where these modifier genes exist, that can be a therapeutic angle as well. I think there's sorry about that, there's web questions.
Well, I have a question in the chat, so I'll start there. This one looks like it's for Louisa. But first, I also want to say, just so that our parents don't get confused, the splice site that's showing different, kind of a different mutation, this is not going to be the same as everyone's splice site. So you have to think of it uniquely. It'll matter where it is on the gene as well. So don't think that all of the splice sites are going to be exactly the same.
Once you've seen a slight splice mutation, you've seen a splice site mutation. You can't generalize it. That's right.
Exactly. Yep, that's a good way to say it. And now the chat question, it looks like it could be for all of you, but for the neuronal and brain organoid studies, have you considered comparing cell lines from different patients with the same mutation? And then what do you think you could see?
I mean, we could if we have cell lines with patients with the same mutations, but do you have a repository where like there are different patients but they have the same mutation?
We are working on that.
Okay.
Yes.
I mean,
we have one variant that is in 5 different patients have the
same variant.
So
Yes.
we're trying to work on getting an iPSC from a different patient.
Sure, I think that would be great because it would also give us clues about how genetic background plays a role in the phenotype that the patient develops. If the phenotype is different, I have no idea about that. Yeah, so.
Yeah, I agree.
Well put.
It would be interesting, yeah, but it has to be available, and these things are really hard to come by. So we have to be happy and glad for what we have.
Thank you. And I think we have another question back here.
Oh, you had your hand.
Oh, there we go.
Never mind, stretching.
Please no movement of your arms or touching your partners. It just confuses us. So hands up if there are questions. So I think maybe it behooves us researchers to come up with our list of 5 to 10 central challenges or main questions that we view as remaining or outstanding from our perspective. And I think informal talks in the next 24 hours among us to start formulating that, I think would be useful, identifying what we know and what we don't know. Yeah, okay, wonderful. Unfortunately, we have no mic. Oh, there's a mic. There you go, okay.
Is there evidence that symptoms become worse or less severe as a child goes through puberty? And if there is, how does that impact the way we're approaching this research?
That's a question for the families, and maybe he's in it. What do you think? Were you listening? You weren't listening. Puberty makes symptoms worse or change at all?
I think we will find an answer to that when we finish our, especially the adult study. That will give a lot of answers. So that's the reason why we really need your support for that.
Have people in the room seen in their kids a change when puberty comes? One there?
Yeah, my
one
daughter's
there?
31, so I definitely, puberty was extremely hard on her.
Okay, yeah.
So it was very difficult, and it was, my daughter's had, I don't want to scare people in the audience. When I talk to people with younger families, this is just my story, so.
How old is your daughter now?
31.
There we go, 31,
So
yeah.
my daughter has had over her lifespan several periods of decline. And for some of those declines, she has been able to get back her skills, some of her skills.
Was
However,
there a clear trigger in any of them?
I would say it was something organically within the brain.
Okay.
The first one was epilepsy.
Right.
The second one was the onset of puberty.
Okay.
And the third one, which changed her radically, was at the age of 21, 22. But menstrual difficulties, she had severe menstrual difficulties, and she changed in personality, but puberty was extremely hard for her, so.
Right, okay.
Yeah,
Well,
that's just my experience.
Right. And yes, sorry, just, thanks very much for that.
I was. My husband and I can speak to a similar experience, and our daughter is 16, and she also went through some dramatic changes just a couple of years ago. So things can get difficult, but they also have gotten better. So things, for us, it seemed like, where did she go? But she came back.
Okay, and what were some of the changes that you saw?
A lot of behavioral changes, and it was difficult at the time to realize that they really stemmed from sleep disturbances. So I'm really advocating for a lot of research behind sleep disturbances and what's happening with our kiddos, because there hasn't been a whole lot of research done particularly for that. And that's why I feel like it's really important for families to get involved in that fundraising aspect and not to kind of shift from a kind of research-based to foundation-based goals. But I think that's really important why families need to get involved in advocacy and fundraising so that we can do more research, not just for sleep, but for all of these symptoms that we're seeing in our individuals.
And did you sort the sleep problem out, do you think, or she aged out of it?
We had to sort through it a little bit, but then, it's interesting when we talk about circadian rhythm too, because she definitely has difficulties with that, and we started to see some of those behaviors and issues coming back toward the beginning of this summer. Right. So we were seeing some of the things that we were seeing a couple of years ago, which ended up landing her actually in the hospital for a couple of weeks. So we realized this is all stemming from sleep again. We have to get things under control, and we were able to curb it a bit, but we don't know where we're going from here. We don't know if...
Right.
So.
Okay, we wait for that paper that you wrote that's going to solve all our problems.
Yeah, I was just going to say one thing about our daughter's changes did not happen during puberty. She's 27 years old, and it started about 25 years old. And we've seen a complete change in behavior in loss of skills and other things and some other medical.
In the last 2 years?
In the last 2 years.
Boy, that's
yeah.
tough.
Yeah, it is tough. Yeah.
Yeah.
So thankfully it wasn't during puberty.
Right.
But it's definitely now. Yeah.
And has she had an EEG or anything?
Yes, and unfortunately, when she's had several EEGs,
Yes.
and we had a 2-week EEG, and
Yes.
that's a perfect condition for her in that nothing happens. You know,
Okay.
we see the behaviors, we see everything else, and at the time, the mobile EEGs weren't a thing, and we weren't able to do. So we
Okay.
may be able to catch it that way, but I mean, it's the perfect environment for her. Nothing happens, everything goes away, and she can have increased behavior, everything before and after, but not during the EEGs. So.
Okay. Thanks for that. And yes.
Was there any trigger of the change? I mean, in something.
She has had triggers, I mean, not in terms of triggers, but things that we saw that symptoms that happened right around the same time were increase in cyclical vomiting for no reason. Her sodium level has been an issue prior to that. Now it's back to where it should be. I mean, issue in that we go into the hospital and have to be rehydrated and everything else. But no, there's not. And we've got everybody on it trying to figure it out. Yeah, our medical team. And yes, low sodium. Low sodium. Same here. Low sodium. Yeah.
Sorry, low sodium associated with dips or just in general you see low sodium?
Crash.
A crash in this case?
This began after the last crash at age 22. We had the low sodium.
Yeah.
And, she takes medication for her sodium now, and
Okay.
it's stabilized, but she does have the issue with low sodium
Okay.
as well. And
Okay.
it's still actually just under normal, but the
Okay.
doctor said that's where she's
Okay.
at.
It
so-
sounds like there's an aha waiting to happen that we haven't been bright enough to have the aha about here. So low sodium observed, whether adrenal crisis or something. Okay, we'll get back to you. Okay. All right. Thanks very much. Collecting microphones here. There you go.
Dr. I don't have questions for our wonderful speakers here. I just want to do a shameless pitch before we break for coffee, which is in 5 minutes, is that we would love to support Louisa and Dr. Werma and Sharon, and we're going to talk about all of that tomorrow, but we can't support everybody without families getting involved. So yes, they want more IPSC lines. Yes, they want all our resources, but we can't do any of that if we don't get families involved in fundraising for us. That's what I want to say.
Tough love. Okay. Wonderful. And this is all about advancing our own careers, but it's nice to hear that. No, I'm just joking. We really feel like we are collectively part of the team, so as much as we can help out as well. Mara.
I can speak to changes in puberty a little bit too. We assumed, so our son is 17 now, and so male, so interesting, different puberty, right? But we assumed a lot of what was happening to him was the effects of seizures and all of the seizure medications we were applying to try to curb those. So, you know, the fact that he was seated because of fatigue, because of sleepiness, and also for safety, because the drop seizures were dangerous and he was very active. So we assumed, you know, the mobility issues that we saw when he finally was seizure-free and we could let him go were all due to that period of time, which Dr. Toomer was mentioning this morning. There's that, you know, that sort of environment issues of just being on his bed or in a chair. But the pronation at his ankles just worsened in the middle of the period where he was mobile and he was moving about and regaining a lot of his mobility. He was getting better at the knees and at the hips, and then all of a sudden his ankles
pronation?
his ankles just dropped, and now he has AFOs. There was a change in the hypotonia. His scoliosis is getting worse.
Right.
So I feel like the hormones did something as well.
Okay.
And then I had a completely different comment. We were talking about measures for epilepsy for mice. I have one identified trigger, and that is barometric pressure changes. And so if you're able to change that in your mice, fall, spring, and sometimes early summer, Cedric is having seizures. He says when he was having regular seizures, he would then have clusters. When he was seizure-free, he would then restart. We were almost 3 years seizure-free, and in last October, at the beginning of the big change in weather where we were, the seizures restarted with a vengeance. And so I
And
feel
if
like
you look
that's
at-
one
if you-
measure.
If you look at the barometric pressure, there are alterations.
Yeah, when there's a big
Yeah.
barometric pressure change,
Okay,
there's
up or down?
a trigger. Yeah, up or down, I guess, because spring
Okay.
and fall.
So I know a barometric sensitivity, epileptic sensitivity is a known thing, but whether it's just Cedric as a one-off or it's more generally found in DLG4.
Right, and
yeah.
it would be interesting, but I think I've mentioned it to parents here and there,
Yes.
and there might be a trend, but it would be interesting
to
to
keep
discuss
people
if
to
anyone
keep-
else
to
has
keep an eye
a
out.
trigger they've identified for
Yeah.
their seizure. Let's
Yeah.
throw it this way.
Yes.
Nice.
Putting it in our zeitgeist, so to speak, yeah. Okay, Sabrina and Mara, we're good?
There's just one more comment in the chat, but this is, I think, to the researchers from Julia Summer from the Simons Foundation. She adds to Katina's talk that there are now iPSC lines for 5 individuals with DLG4 variants available. 2 of those iPSC lines are from individuals with the same variant, so you should be able to get those. All lines are listed, and we can send you the Google Sheet for that. There is also phenotypic data available for those donors, as Katina presented earlier, and all data and biospecimens can be requested through Safari Base. So if you're interested, I can give this to you as well.
Wonderful.
And
Great, okay.
I just want to say that, I also, in my talk, I said that one of the questions in the chat was asking for the splice variants. Most of the splice variants, we expect that they will affect transcription. But I agree that all the splice variants should be, indeed all the variants, also the nonsense and friendship variants, it's a good idea to check them with RNA studies. I completely agree with that, and if you do that, we have good experience in that. I will guide you and help you in any case and in any way. We can also carry out these without any, asking for anything for support. And for this, it is better not to use blood RNA. It is not very useful, but isolate cultural lymphocytes, white blood cells, lymphoblastoids, and use them, or skin biopsies. So if you get only the RNA blood in a PAX tube, it's not good for, it doesn't give very good results, because we use these experiments for diagnosis, and our diagnosis is completely, under control, and everything is with the quality control. So these are very important things, and we will happy to both support the families and the researchers with any of the experiments when sharing everything.
Dr. Huber, would it be RNA, would it be useful with nasal?
No, it's not very good. And this is the other thing, is that we also have controls for both the fibroblasts and the culture's lymphocytes, because we kind of transform them and immortalize these white blood cells, and the immortalized cells are very stable. So they give different results. We have important results with that they are different.
Need, like skin biopsies from
The skin biopsies will be the perfect, and it's so easy to also send, because you don't need to send it in freezed or nitrogen. You can, but I know it is not very easy. But on the other hand, there are now punch biopsies which doesn't hurt that much. No. So I will recommend doing that. And DLG4 is better expressed in the fibroblasts compared to the blood.
Really? Okay.
We have a higher expression, so we have better results with the skin biopsies.
Then
so
with
this information I want to share with everybody.
So better than EBV transformed
Yes, exactly.
lymphocytes,
Yes.
skin biopsies best.
Yes.
And so just to be clear, as we researchers are fussing about whether the protein's up or down, what Zainab was asking about, never mind the protein, let's just look at the RNA, which is the intermediate
Start with that
step.
at least. Start with that.
And then you may get the answer with the RNA. If the RNA just isn't there or is dropped, then we don't need to keep on asking questions about the protein. We have our answer. I think that's what you're saying.
Rami.
Yep, okay.
You just stay there.
Okay, stay there. I do what I'm told.
Yeah. What I would suggest is that since our cell of interest, and we want to believe that this is a neuronal phenotype, is that there should be some kind of comparative studies for RNA that will compare, in the case like you're suggesting in the fibroblast, at least to the neurons, and making sure that there is compatibility or redundant or concordance between how it is regulated. In our hand, and I will share some data, the RNA is not predictive of what is happening in the protein. And I think that there is multiple factors that are regulating this very important molecule that could also result from context like neuronal versus fibroblast. I did not do the fibroblast, so I don't know, but I just would suggest that there would be a comparative standard at the beginning just to compare what is happening both at the RNA and at the protein level between fibroblasts and neurons, so you can consider this as an easy readout for the future.
Okay.
What we're trying to do is we have done this comparison with the fibroblast blood cells and cDNA, both from the what's called commercial. So, but if I completely agree that you have to. So what we do is that this is, less expressed in the blood, but we didn't do of course, for the patients, because then we will need neurons which are developed to look at that. But I think I agree with you that it's a good idea. The only thing I'm saying is that from a diagnostic point of view, I'm a clinician, so for me it is very important that the research is very important. Of course, it's very important, but at the first line to understand, is it pathogenic? Is it really the cause of? That's my main starting point and concern. So that's the reason why I say I will help anything for the RNA, but I agree with you that we should do that. And I will, if there's somebody having all these cell lines form single and for a couple of, together with controls, and I will, as I say, share all the protocols.
Rosie in the back.
Rose in the back.
I'm running.
So that's my job.
My question's probably overly simplistic, but everybody today kind of really talked about how DLG4 impacts the PS95. Are there other genetic issues, like other syndromes, that also are impacting PS95? And then how do we get smart about those and see what the correlations are or shared findings to kind of help with therapies with our kids?
One of my dreams and what I really want to do is to look at some of the, especially proteins or genes which are talking really very much at the postsynaptic region, some of the receptors, and there are some proteins talking with. I show a picture of our collaborator from Holland, Heske. So for example, she's working with a protein which phosphorylates, gives a phosphor to all the proteins. And there are some neurodevelopmental disorders, SYNGAP, SHANK, they share common things. So there are different ways of doing this. One of the things I want to do is to see, for example, take a person with a PSD95 mutation and then see how the other genes are expressed. And I think that's the reason why the thing you are going, the RNA-seq is a good way, though it's not very specific. So I prefer RT-PCR because you have a good result. But choose some of the, okay, do they change the expression of these? But probably they do, and they affect each other. Especially some proteins like PSD95, CAMK2, they affect many other proteins because they are talking so much with the other proteins.
Yeah.
And also we want to compare the clinical phenotypes of different neurodevelopmental disorders with each other to see what are different. Can we correct one of them or make one of them better so that the other affects? So these are things we really want to do. I agree completely.
A number of genes in Simon's searchlight are in the same, as you know,
Yeah,
in that neighborhood, Shank and others. Yeah. So good question.
Dr. Tewart, just quickly.
So we can adapt your protocol for RNA in our iPSC-derived neurons as well? Okay, okay. Yes, exactly. Yes, yes. And I forget your name, I'm really sorry, but I know Rami, Dr. Rami. So I know another protein mutation, CHD2 mutation, that's a very classic example. Everybody sees CHD2-associated epileptic encephalopathy. Everybody sees that there is an increase in RNA with the mutation, but there is a decrease in the protein levels. Like not just one lab, every single lab that works on CHD2 sees this phenotype. So I totally agree that just the RNA readout might not be enough to look at the, we need to see the protein expression as well. Yeah. Yes, chaser, not long, not coding RNA.
And also there's something else. Genetics, I say genetics is very complicated. Some of the nonsense variants, which makes a stop in the beginning of the gene. Yeah. Some of in some mechanisms that when the protein is made, they just doesn't see that, close the eyes and then make the protein. Yes. So there are so many exceptions. But we have to start with the generals.
Right.
So that's
okay.